I have a couple topics I'd like to talk to you about today, tell you about. First, I have two slides about Dipharma, if you're not familiar with the Dipharma group. And then I have basically three topics for you. I want to tell you about how we manage risk and innovation. So I have three chemical examples. So for those of you who are chemists, it's good. For those of you who are not, I'm sorry. The next topic I have for you is solid state development. I want to talk to you a little bit about how we manage that in the group. Again, for us, it's mostly in the lens of risk mitigation. And at the end, I have one example of how we use the same type of tools, the same type of approach outside the lab when we're managing business cases. So how do we manage global integration? How do we manage projects that are large and complex? So Dipharma is a large group. We manufacture both API for the generics market and the CDMO market for new chemical entities. We do that from two R&D centers, one in Kalamazoo, Michigan, where I'm based, and the second, which is in Barnzate, which is 2 kilometers from where we are today. So it's a very convenient show for us here in Milan. The other two sites are in Corona, which is 20 kilometers from here, and the Moreto site is north of Venice by about an hour. The other thing I wanted to point out here, and it has already been mentioned, you can see FDA inspected in Kalamazoo in 2024. So that's brand new for us, very excited about it. But the inspection record on the Italian side goes back to the 70s. So that is basically the second box here. I wanted to just point out it's not just the FDA. We work with all the global regulatory agencies. We've been doing this for 75 years. I'll talk about that a little bit more in a moment. And then the last two is where this all ties together for us, because the experience and the risk mitigation comes from the people that we have. More than 90 professionals in our R&D centers, again in Kalamazoo and in Barnzate, not far from here. And then the 600 other individuals, the other 500 individuals that make the rest of the operation work. And the picture down below is of our facility in Moreto. If you look closely, you can see the Alps in the background. Okay, so I don't think I need to spend too much time convincing you that manufacturing APIs, chemicals, is inherently risky. Right? We hold a lot of energy, whether it's in solvent or in the materials that are in the reactors, and that has to be properly managed. If it is not, it can have large consequences, either for patients who lose access to a life-saving medication, or for our partners who can't get that to market. That has an effect on them as well. And then the other two are a little bit more acute, right? Because employees can be injured. Deaths occur in plants. And I don't know if it's a big deal in the United States. I don't know how much it's been watched here, but Hurricane Helene just made landfall in Florida a few weeks ago. And there was a facility in Conyers, Georgia from Biolabs that ended up with a 10-kilometer exclusion radius around it as it burned. So the communities near these plants can also be dramatically affected when these things go wrong. Now, we manage high energy chemistry. And so the risks are inherently higher. And so we need to manage that in a slightly different way. So I've got a couple examples here of the types of chemistry, and I'll show you a couple of these. The first one is alkyl nitrites. They're high energy species that they're very useful for CN bond forming reactions. Nitroglycerin is a cardiac API, but if you're also not familiar, it's the main component, it's the explosive component in dynamite. So that needs to be handled in an appropriate manner. And then a couple of examples now with some structures, candesartan, rufinamide. You got the tetrazole moiety here, the triazole moiety here. Those are all high energy. Whenever you see nitrogen atoms together, you need to think about that. And then the other ones we use are just azides in general, which I've illustrated here in this acyl azide in the pramipexole synthesis. So I'll tell you a little bit about these processes. So starting with the simple one, I'm not going to go through this in much detail, but the alkyl nitrite is up here at the top. We make it from an alcohol, some acid, and some nitrite. You mix in a ketone, and you end up with the alpha-ketoxime shown. Now, this is what we want to make, but we need to be able to manage this process. Why are we concerned about this? Well, you can see this picture here. This is the DSC trace. And the details are not important. What you need to know is that it's a very sharp peak, and it's very big. At 200 degrees C, this compound emits 2,230 joules per gram. That is 48% the energy content of TNT. So again, something worth some respect. In this case, it's even a little more interesting because alkyl nitrites are also highly toxic. They are vasodilators. So as they enter the system, your cardiovascular system increases in volume. Your heart has to pump a lot faster, harder to keep your blood pressure up. If it can't, you faint. If you can't recover from that, death can occur. So this one is really interesting because we have a double risk here. Not only do we have the risk of managing the energy, but we also have the risk of managing the potential health impacts. Okay, so I hope at this point we can agree you want to know something about that. It demands specific knowledge and it also demands specific controls, and I'll give you a couple of examples of that as we step forward. So I won't go through it in too much detail, but this is the kind of schematic of the reaction. You've got a reactor, you add the nitrite, and you make a whole tank of this alkyl nitrite species. That is the highest energy you can have, right? You have a tank full of flammable solvent, full of an explosive compound, and this is what we would like to avoid if we can, right? So we want to avoid this, and the way we do it is very simple. It's by making this a continuous flow process. So schematically shown in the bottom left, the acid and the alcohol are mixed. The nitrite comes into a small reactor where the reaction occurs. There's a pressure regulator, a phase split, and the alkyl nitrite comes out the side. Now what we do is what's showing up here in dosing. Instead of making a tank full of this stuff, we use a small volume in the reactor with the ketone, and then we dose in the nitrite at the rate at which it is consumed. So you form a steady state concentration, but the amount of this compound never builds up with time. Then, when we're done, we can quench, we can remove, and we can move on, just as we would normally. Now, the nice thing about that is it completely removes the energy. But for those of you who have seen these kinds of flow processes developed in an R&D lab, this is usually what they look like. It's not very complicated, but there's a lot going on. And what the expertise in our background allows us to do is move much more quickly into plant. And one of the keys for us is the logo here in the bottom corner. Biazzi Engineering is an affiliate of ours. So they're a member of the greater Dipharma group. And they build plants. And so we work with them in an engineering sense quite often to implement these types of processes. So it allows us to very quickly transition from the organized chaos you see on the far side to the nice, neat, clean plant. So I said that we have a lot of experience doing this. So I want to talk about that for just a moment here. Mario Biazzi is our founder. He patented a continuous flow process for the production of nitroglycerin in the 30s. This is the technology that the Dipharma group was founded on in the late 1940s. And so you can see the schematic of the reactor from the patent, and you can see on the right-hand side here a photo of the plant that we currently use to produce nitroglycerin. And so to this day, we are using this technology. So we have 75 years of experience in this new technology of continuous flow chemistry. All right. Move on to a slightly newer API because nitroglycerin has been around for a very long time. Candesartan is showing up in the top right. And what we need to do here is take this nitrile and convert it into this tetrazole here. It's plant chemistry, right? So it has to be robust. It has to be scalable. In this case, we need to do this on metric ton scale. And so we're going to require here one of those azide sources that is safe and in sufficient quality to allow us to manage this. Additionally, we want to make sure that we have a way of controlling the azide at every step of this reaction. So when it comes in, and every possible place it could be along the way. And that's to the bottom 2 points. We need something that's environmentally conscious. We can't be sending this stuff out into the world. And we need something that's very specific and very sensitive for the measurement of these types of compounds. Now, I want to take 1 minute and talk about this. You can see 2 news articles on the left-hand side, explosion with sodium azide, azide explosions discussed further, and a picture of a hood from the University of Minnesota in 2014. This is my alma mater. This is something I watched very closely as it occurred. There was a student that was actually very badly injured here, and thankfully they recovered. But working with azides can have a lot of risks, and one that I want you to be aware of in case you're not is the formation of hydrozoic acid. You should assume, if you ever use an azide, that you're forming hydrozoic acid. Now, the problem with hydrozoic acid is it's an inhalation poison, but second of all, it is itself an explosive. And it's a very volatile liquid or gas. It boils right around room temperature. It doesn't require any oxygen to detonate. So you can detonate the headspace of an inerted reactor. Okay? So you have to always be careful about this, and you always have to be aware of this. So the methods that you create and the controls that you put in place have to account for this. So the easiest way to deal with azides is given away at the top here. We're going to increase molecular weight. In this case, we already knew that tetrabutyl tin azide is a great reagent to do this kind of work. It's kind of borderline in its energetics. So the easiest way, the simple answers are usually the easiest. They're usually the best. And in this case, the simplest answer is just to increase the molecular weight of the azide species. So you go from tetrabutyl to tetraoctyl, you increase your safety margin by a dramatic amount. The problem is that tetraoctyl tin azide, while it is available on the commercial market, it is not available in the quantities at the required quality to affect the synthesis inside the GMP plant. So we took to making it on our own. So this is an in-house product of ours. I'm not going to go into too much detail, but you can see one chromatogram here. This is the chromatogram of hydrozoic acid measured in the headspace of a reactor at 0.4 parts per million. So we can measure hydrozoic acid content and azide content in general down to that kind of a level. So exquisitely sensitive, nice controls, and this method has been proven out in many different manifolds, that one that we can use to assay many different, the headspace, the reactor, the waste streams, etc. And we use this routinely to ensure that we have the proper controls as we need them. I'll come back to that on the next example. And something that I'm going to mention here, but not get into the detail of, is that this is a route where we have nitrosamine control at every single step, not just in the azide step. We can talk about that offline if you'd like. Another example is rufinamide, triazole. I showed it before. Here we have a lot of problems, okay? This alkyl azide in brackets is an incredibly high energy species. Alkyl azides are usually very bad. The smaller the molecular weight, the worse they are. We have an alkyl propylate ester, which is also a high energy compound. It's not explosive, but it's also high energy. And we have copper sulfate. Copper sulfate and sodium azide makes copper azide, which is a known explosive, and it's used as an explosive. So this is something that we definitely don't want to be forming. Again, if you don't have much experience with azides, just think of any azide that you hear. Any metal azide other than sodium azide is probably an explosive. Okay, so we have these things to deal with. Based upon what I told you earlier, I think you're all thinking that I'm going to predict that you're all thinking, okay, this is going to be a flow process, that we're going to fix this with flow. The problem is we can't. Not every process is amenable to flow chemistry. There has to be specific kinetic parameters to make it work. And in this case, through a lot of study, don't think we didn't try, but this does not work as a flow reaction. And so we actually run this in batch. And again, now, since we are creating a large amount of these species, we have more controls in place to deal with them. So back to that method I showed you, if you think back to the 0.4 ppm, that's the same method that we use here to monitor azide. We have a specific IPC in place. And at the end of the sodium azide reaction for what is carrying into the copper sulfate to prove that there is no that the carryover of the azide is acceptable to the amount of copper. And there's a specific safety evaluation that we go through whenever we have an azide and a metal, and we do that here too. Okay, so enough structures for a little while. I have a couple other pictures for you, but again, just to start off, solid state development. Why is this important? I think most of us are familiar with this as polymorphism in an API, where you change the biological activity of a compound based upon the physical form that it's in. There's been a couple of APIs that have had issues with this in the past. It is also something that is worth monitoring throughout your process, because the physical form of your intermediates can affect their solubility, the way they dissolve. Right? Solubility is a thermodynamic property, dissolution's kinetics. You want to understand both of them. So just very quickly here, if you look at the solution on the left, this is what we want to see. It's a nice suspension, looks like paint. You don't see any clinging to the sides of the beaker. You look at this one over here, it looks like ricotta, right? It looks like cottage cheese, and you can see how much of it is stuck to the walls. This is not what we want in a plant. This is not going to move. This is going to be a disaster. The real problem for us was, as we started development, we started over there, and the further we went, the more we ended up over here. So let's explore that a little bit. So the first thing we say is, okay, maybe we have a different particle size. So the particle size distributions are shown on the left and the upper. They're almost identical. In the middle, we have the XRPD to show the polymorphic form. It's a match. The only thing that we could find the difference is this little difference in the baseline here. A little difference in the background. So we went and explored that a little bit. So if you look here at the top, you see that green line, that flat featureless line. That is the amorphous form of this material. And so what we learned is the materials that we liked that filtered well were highly crystalline, 83% crystalline material, 17% amorphous. The materials that didn't crystallize were less, or didn't flow, were less crystalline. They were more amorphous. So now we have a testable hypothesis, right? We think we know what's going on. We have an assay to check it. So now we have to figure out what variable we need to control to ensure we're going to get what we want. In this case, again, a very simple fix. If you keep the distillation, the temperature of the distillation during the crystallization above 24 degrees, everything is fine. We'll be able to maintain the crystallinity we want and stay out of the area where we're going to form the amorphous material. Okay. So now I'm going to switch gears and talk about a little project management, a little business, more about how we manage the programs we do. So I guess I'll start by saying a lot of people feel that when you deal with high energy chemistry, you have to be slow. But what you need to be is disciplined, deliberate. Right? You don't need to be slow, but you need to be disciplined. Those same ideas follow over here as well. Right? Complexity in development, complexity within an organization, within a program, can all be managed the same way. Okay, so as a case study, we were asked to develop a 7-route process, including a very challenging, unique fluorination step from a US-based multinational company. And we needed a timely supply of 2 non-GMP lots, one for development purposes, one for toxicology, and a first-in-human phase 1 lot. Now, you can see the challenges here because you're probably reading faster than I'm speaking at this point. The project was initiated during COVID. I'm sure you all remember this. I'm sure you all remember what supply chains looked like. I'm sure you all remembered what it was like to try to get things done during that time. But on top of that, we also had what was a rapidly evolving clinical program. like I said, we haven't really gotten to toxicology yet. We really haven't, but we're already working on first in human. And so during the course of this program, the material needs change rather dramatically. Okay, so what we have are three things that need to be managed here. We need to manage that rapidly evolving clinical development program. We need to manage supply chain. And we need to manage the communication, the handoffs, the project management side of things. And that's where the discipline and the deliberate nature comes back to play. So what happens when you're having issues, knowing what the outcome of what the value of material you need to make, how many kilograms you need to come out with, you oversource your materials, right? You protect yourself. In this case, because it was during COVID, we double sourced materials in double the quantity. That was fortuitous for us because the change in the request was doubling the output. So then we weren't oversourced by 4 times, just 2. Unfortunately, we also had, even though we had double sourced everything, one of the materials was not delivered to us. And of course, that never happens when you want it to. It never happens 6 months in advance. This was just a few weeks in advance. So at this point, we no longer have the ability to really find another vendor. So we looked for, the question we asked supply chain was, what can you find us that's in the route to make this compound? And they were able to find the materials that we needed 2 steps back and secure them from a local source so they could be delivered within the week. So that's perfect. We had to turn around to planning then and say, okay, where can you find us the space to get this done in the next 3 weeks? And so they moved things around and found us the room in the Corono site, which was just up the road from here. But this is now the 3rd site we're using to manage this. So it's feeling a little complex. That's where the rest of this comes into play, right? That's where the discipline comes in. And that's one of the things you find is just part of the culture at this point. We have a single unified view of our project teams. So we're using 3 sites, multinational organization. They have many moving parts as well. And so we've developed specific tools and methods to communicate. But it's, suffice it to say, rapid and concise, both internally and externally, to make sure that we're managing across, that everybody is informed. And we have a unified view of the program, right, which allows us to actually kind of take advantage of time zones, because the work that's happening in Kalamazoo can inform what's happening here in Italy. Italy then can inform Kalamazoo before they get up what they need to be doing. And so we can kind of decrease our cycle time that way. So it sounds like in some cases that these are disadvantages, but if you look to see how you can use them and you approach the program in a disciplined way, it becomes an advantage. All right. So, bottom line, the complexity that we ended up with was a delivery from Corono on a 15 kilogram scale, plus the non-GMP KSM that was required to feed into the Moretto facility, where we made the RSM and the tox lot. The RSM came to Kalamazoo, where we finished off the first in human phase 1 material at the different scales required. And everything in this case was delivered on time and in full. And we had a party when we were done. I mean, we deserved it. All right, so I hope I've given you a flavor of how some of the kinds of approaches you can take in the high energy chemistry can tie into non-laboratory activities, and why you might want to think about all the things you do in a risk mitigation sense. And thank you for your attention.